Welcome to the Doctors Making a Difference podcast, where we help physicians to be empowered with the tools they need to be successful in medicine, in finance, and in life. Join us as we highlight doctors and other professionals around the world who are making a difference. Today we have the privilege of having Heather Hayenga. Heather has a unique story and is a unique person. I'm really thankful that she was able to come on the podcast today and talk about her story. She is a PhD researcher at the University of Texas and has an interesting connection with my story and with so many other folks who have cancer and research cancer. I think you'll be really interested to hear Heather's story, and I appreciate you coming on. So Heather, would you mind telling the audience about yourself? Introduce yourself a little bit.
Sure, yeah. I am an associate professor of bioengineering at the University of Texas at Dallas. My research focuses on cardiovascular modeling as well as cancer for the last 5 years, because I unfortunately developed a rare cancer, solitary fibrous tumor, that originated in my brain back in 2013.
That's a brief introduction to a very complicated, long story. Tell us a little bit about when you were thinking about what kind of work you wanted to do in college. How did you decide on medical research and specifically cardiovascular research? That's where your initial focus was.
I am really driven by hardships, I guess. I think hardships drive motivation. And so, unfortunately, my father, we were playing basketball and he passed away of a heart attack, and I couldn't bring him back. And so that was devastating to me. And the next week, I joined an immunology lab and I wanted to start working on cardiovascular research. It's a disease that has been the number one cause of death for over 100 years, and we haven't come up with a cure. It's still the most prevalent disease, and I just thought we have to change that. That's how I got into cardiovascular research, and then I went on to graduate school, and I was very intentional and said I want to work on cardiovascular research. And my advisor at Texas A&M, he said, that's not something we do in the lab, but I have some discretionary funds if you have a clear idea. And I was like, yeah, I do. That would be great. And I got to use sort of modeling techniques and grow mice with plaques and see how mechanics affects plaque rupture and stability. And now my graduate student just graduated, my fourth graduate student. He's going to create a company that can go into the clinic, and it's going to be a cardiovascular tool that can predict plaque rupture as well as what treatment would be best to treat atherosclerotic plaques in people. So I'm really excited that he's bringing that to fruition. All of those 15 years of work, yeah.
Oh, yeah. How old was your dad when he passed away? Having passed away so suddenly while you were playing basketball would have been just devastating.
Yeah, I think I was 24 or so at the time, and he was 52. So he still had a long life ahead of him. My brother was still in high school, and after he graduated high school, he was planning on traveling and, yeah.
Oh, yeah. 52 is really young, especially by today's standards. The average age that people live in the United States is well into your 80s, maybe sometimes late 70s, but to suddenly be gone at 52 is crazy. I have a lot of cardiovascular disease in my family as well, and it is crazy. So I can see why you were motivated to do that. And it sounds like you spent your graduate school years focusing really a ton on that side of research. You've had people that have done that, and it's been a successful journey. But you pivoted. Unfortunately, like you shared at the beginning, solitary fibrous tumor is a disease that you and I both unfortunately have. Obviously, that affected your life. And so do you mind describing that process of pivoting from cardiovascular research to oncology research?
As an engineer, I like to find solutions, right? When I found out that I had this cancer, I did a lot of research, and I found out that the oncogenic driver of this cancer is a fusion between two genes on chromosome 12. So basically, NAB2, which kind of suppresses proliferation, things like that, and STAT6, which augments proliferation, they work together. So one is kind of a negative feedback and one is positive feedback. But in this cancer, unfortunately, they fuse together. And so that repression never happens. And so the feedback loop stays on. And so I thought, okay, this is a genetic mutation or fusion, aberration. Let's just use CRISPR. Let's reverse it. Let's put a kill gene in there or something like this. And there we go, problem solved. And it will be very specific to like the CRISPR will find the NAP2-STAT6 fusion, and it will only edit the DNA of the cancer cells. So UT Dallas is a very collaborative environment, and so we had a genetic engineer professor across the bay. And so I told him about my diagnosis. I don't display that I have this cancer very often, but I told him about it, and I said, what do you think? Can we use CRISPR? And he's like, I don't know, but I'm willing to find out. We started in the Petri dish, and unfortunately, we didn't have any of these rare cancer cells because I had brain surgery and they fixed all the tissue, and it's so rare that I couldn't get cells. So first we had to engineer the cells. So first we had to put the gene fusion in cells, and that took about 6 months because it was pretty hard to do, transfect parasites and the cell type of thought to be the cell type that this cancer is derived from. So we put the gene fusion in there, and then we designed a CRISPR enzyme to target it and edit it, and it was pretty successful. It reduced the proliferation rate significantly by 60 to 80%. So we're like, great. And then we were teaming up with other people for drug delivery approaches. But unfortunately, CRISPR has not really been used readily in the body systemically. And that is because for delivery to solid tumors, the efficiency is so low. If you can imagine that even if you put it in the IV, it has to go to the tumor cells, the CRISPR enzyme, and then pass the membrane, and then pass the nuclear membrane, and then go into the DNA and edit it. And we in the lab, we can use transfection agents and things like this to help put a high concentration right on the cells. You can't do that in the body. So that was sort of our roadblock of that original idea. And we're still kind of working on it.
So it was a kind of a delivery system, or the ability to deliver that CRISPR gene into the actual affected cells deep within a tumor was a lot harder in vivo than it was in vitro, because you're dealing with all those layers of cellularity and just membranes and a lot of things. So that sounds like that was a brilliant idea. And I'm assuming since then you've been able to shift and try other things. What else have you been able to try in your lab?
Yeah, so we're still working on adenoviruses and other ways, nanoparticles, to package the CRISPR enzymes. But then we thought, okay, what if we don't edit it at the DNA level, but more at the RNA level? Then it has to just cross one membrane. So we designed a CRISPR-Cas13 to target the RNA level, but still the deliverability was a bit challenging. So then we pivoted to antisense oligonucleotides, which are mRNA sequence of mRNA, and they bind, like a lot of the COVID therapies were mRNA as well, but mRNA basically binds to the gene fusion, the NAP2-STAT6 gene fusion, and will block it from being translated into a protein. And so that showed pretty good promise as well. And so we're still working with Ionis in California to develop ASOs, and we did an animal study with the ASO, and it slowed the proliferation in SFT tumors in mice, but didn't exactly kill them. So it will just slow the growth, but it didn't kill them. So the gene editing, the ASOs, and then we tried immunotherapies. A lot of cancers are driven by a limited amount of gene aberrations or mutations. And so because this cancer is really only driven by one mutation, we thought that this one mutation can be translated into a protein that can be chopped up, and little bits of peptides expressed on the MHC class 1 complex. And then T cells will come and recognize that. If we develop antibodies, modify the T cells, then we can use an immunotherapy approach and have T cells targets these SFT cells specifically and eliminate them first we have to see if there is a little peptide, unique peptide on that MHC complex. And so we ran mass spec on the SFT cells that we had in the lab. At this point, we had some donor samples. We have 3 patient-derived cell lines now, in addition to our engineered cell lines. But the mass spec wasn't sensitive enough to detect the peptides. So we're in the process of looking for more sensitive mass spec in Germany, teaming with them to develop a more mass-sensitive detection method of these unique neoantigens or peptides. So immunotherapy is our third therapeutic approach. And then the other one that was funded by R01 just last year is repurposing of drugs. So we have all of these cancer drugs, right? And 2,600 FDA-approved drugs were specifically screened on these patient-derived cells and engineered cells to see if any of them selectively would kill the cancer cells, but not the normal cells. And so we had a hit. We had about, 20 hits or so. And so we're looking at the most promising ones right now, which is mevabrasib and some other ones that may be able to be repurposed, have a better chance at treating this cancer.
I just want to thank you for what you're doing. It obviously impacts you and your life and your family, but all of us that have solitary fibrous tumor are very thankful for you and your ability to understand it. And you're right in the middle of it. It's interesting. I think people are placed where they need to be for a reason. And I think that's quite interesting. I would never wish solitary fibrous tumor on anybody, but isn't it interesting that a PhD researcher who knows how to run clinical trials and do all this stuff has this diagnosis, and it's not just an abstract interest. You're very interested in it because it affects your life and your family. And I wanted to ask you about that. How has that experience been to be diagnosed with solitary fibrous tumor? You shared with me that you have children at home and you're in that phase of life where things are so busy, and then to still be working and living the patient life, but at the same time having to go and research it and try to research your own disease.
We all juggle, right? Being a full-time doctor or a full-time professor, as well as mom, as well as patient. So the department has been super supportive. And so I think that's really helped changing my research in my lab to also look at SFT cures. Kind of allows me to look at treatments for myself and do research for that while still publishing papers and getting grants and things like this. So I just found a way to merge the two together, doing research that's impactful while also helping pave the way for treatments for myself and understanding the treatments. Because I have not taken the conventional approach, the standard of care approach. I know a lot of doctors like to take the protocol treatment off the shelf, which is chemo and systemics. But some doctors are willing to just think outside of the box and be open and take this journey with me. And so I'm working on my third case study with my liver ablation doctor. He said, I've treated 119 tumors in your liver. That's a world record. That is,
oh, wow.
No one in the world has had that many tumors treated by ablation.
Oh.
This has happened over 5 years, and he was willing to take the journey with me and just treat 6 tumors every 6 months or so. And then the liver is, fortunately, the only organ that I know of that can grow and regenerate. And so we've just been slowly combating these tumors. So he's going to write a case study to show that maybe going straight to drugs is not necessarily the only approach. Another oncologist at MD Anderson, he wanted me to go on drugs in 2019. I said, overall survival is 2 years, and I don't like that. And he's like, okay, you can try a different approach. And so I tried this sort of local approach through surgery and radiation and ablation. And 2 years later, I came back in 2021, and he was like, wow, you don't have any side effects, you don't have any pain. And he was just so impressed. He's like, if you were to go on systemics and look this good, I would be so happy. I would be celebrating right now. I would be saying, good job, I did something good. And so he's like, I don't have any systemics to recommend to you right now because whatever you're doing is working. And so I think you have to be brave and courageous. And I don't know what I'm doing, but like I've had good mentors in my life and really, really good surgeons and good doctors. We've taken the sort of unbeaten path.
Oh, yeah. You told me just before we hit record about histiotripsy. I've noticed that some cancer centers can do it, some can't. But do you mind telling, not everybody on the podcast will know what histiotripsy is because it's a little bit newer. Do you mind walking us through what that is and how you've used that for tissue ablation?
Yeah, so it was developed at the University of Michigan, then went to Europe and clinical trials there, and it had very good rate of success. So I was the third patient in the US to try it when it came for clinical trials here. And it's basically using ultrasound waves to cause these microbubbles at the tumor location, at the tumor site. And this rapid increase and decrease in the microbubbles causes mechanical deformation, and it ruptures physically the cell membrane and the cells. And so they're thinking that there could be some immune scopal effect because all the cancer cell bits are released and immune cells can target that and potentially target other cancer in the body. I didn't have any immune response, but the 3 lesions that were treated in the trial 2 years ago have been disappeared and gone. And it's really nice because histotripsy, you don't have to make any incisions. You just put a water bath essentially on your stomach or wherever you're treating, and then put the transducers on that. And the interventional radiologist determines all the physics and things like this to modify the transducers. And I wake up from general feeling like nothing happened. It has very little side effects. I think it's a promising technology, and Histosonics, which is the company name, just got bought out by a whole corporation of people. And so like it's moving forward, really developing and getting better. Dr. Henshaw, the ablation doctor at UW, said eventually it'll be used for more things than just the liver. They could treat cancers within 14 or so centimeters of the surface of the skin, in the brain, other places. I'm staying in tune with that technology.
Oh, yeah. So right now it's just approved or studied mostly in the liver, but has a potential to help with many different tissue types. I'm sure it's dependent on the ability of the tissue to carry sound waves because of the nature of the treatment. I know I inquired about it a little bit on lung metastases, and that's one of the tricky things because there's so much air within the alveoli and the lung tissue. It would be a little bit harder, I think, to get really reliable results, but I don't know. It's interesting to see how far that technology can come and how much it could change the way we treat some of these relatively rare or slower growing type of cancers.
Right, yeah. The one deep in my liver and more towards the top of the liver, so they actually did have to go through the lung. They weren't successful this last time a couple of weeks ago in actually treating the cancer itself because of the sound wave going through the ribs, the bone, the muscle, the lung air, and you could just imagine each one of those is a different material. And so sound waves travel differently, and so it's really hard to get them to meet up at the tumor site. Exactly. It's better with homogeneous material types.
So Heather, you're a cancer warrior. Not only do you have this and are treating it as a patient, but you're out there fighting the fight for all of us that have cancer. And I think people listening to this, there might not be too many people that have solitary fibrous tumor, but I'll bet somebody listening to this either has it or has a patient that has it. As physicians, like I know about sarcomas, but as far as solitary fibrous tumor, if I had ever read about it, I didn't remember. It was such a rare cancer. That was another question I was going to ask you. On these super rare cancers where you're talking about 1 in a million patients and the sample size on most of the studies are super low, where do you see that going? Like, what do you think would make the biggest difference for us as physicians or researchers when we're talking about these rare cancer types, because in some ways it feels hopeless because there's so little information. And yet you've outlined a whole series of things that with the right tweaking and funding and adjustment could really work and go from the world of systemic therapy to a lot more focused immunotherapy and things of that nature that could really move the needle on this. And I'm curious what your thoughts are on rare cancer research in general.
Yeah, solitary fibrous tumor is zero point 0.6 in a million. But there's what I think I heard 7,000 rare cancers or diseases or something like that. The NLORM Foundation is working on treatments for ultra rare cancers and diseases. So I think it's important, even though it's, just affects a small population, of course, funding and the government and stuff like this, you have to make these decisions. Are you going to treat cardiovascular disease that affects pretty much a lot of people, or provide funding for SFT research that only treats a handful of people? So what I find is that we can establish a technique or an approach, and cancer in general is driven by oncogenic mutations or alterations in the DNA. And so a lot of our therapy approaches are focusing on understanding these mutations at the genetic level. And so if we could develop a treatment that combats that mutation or whatnot, then theoretically it could be brought more broadly to other cancers. That's my hope, is that if we find something, then not only will it affect a small group of SFT patients, which alone is important, but can also affect more broadly other people by applying the same approach.
And as we understand the molecular biology and the understanding of what signaling mechanisms and what DNA mutations are involved, I mean, there's a lot of hope because, like you mentioned, CRISPR technology and messenger RNA technology allow you to get right down to that signaling level, like you say, either in the nucleus or out in the cytoplasm with the messenger RNA to determine, instead of just broadly hitting all cancers, stopping all vascular growth like we do with Avastin, try to get a little bit more specific, whether it be immune therapy that we're delivering through messenger RNA, or like you say, if you can express some of those peptides or some of those antigens on the outside of a cell and trigger your own immune system to go and fight the cancer. Those things are fairly broadly applicable, not just to solitary fibrous tumor, but you could see those same principles being applied, and almost like a tailor-made approach for an individual with cancer. If you say this is your cancer with your DNA, not just that you have breast cancer, colon cancer, whatever it may be, but you have this specific thing, and that level of specificity. From everything I've read, I think that day is coming. For those of us that have cancer, I want that day to be here now.
Yeah, and one patient-specific treatments, I think that is where we are headed as well. That is going to be the future. And that's with my cardiovascular work too. It's patient-specific modeling, because cardiologists have to make these hard decisions on how to treat someone's cardiovascular disease. Is this plaque going to rupture? Do I need to put in a bypass? Do I need to put in a stent? And one cardiologist told me that insurance typically doesn't fight or give us any grief if the plaque blocks the artery by 70%. And then we could just go put on a stent. But if you look at autopsy studies, a lot of times the larger plaques, like the 70% plaques, are more stable and they don't cause any damage. It's those sort of new plaques that are developing that are unstable, and those rupture more often. Our modeling technique can put all the imaging data into a meshing algorithm and finite element analysis and hemodynamic analysis, and really create a patient-specific model that tells them the vulnerabilities within that plaque. So, yeah, I think that's what I want to see is individualized treatments and care, because we're not all the same.
No, we're not. We try to think of diseases. I'm a primary care doctor, and so the training is to think of, you have diabetes or you have cardiovascular disease or you have whatever. But like I'm thinking diabetes is another good example of, there's so many different types. We don't break it down into subtypes, but I can think of hundreds and hundreds of patients I've treated with diabetes, and they react so different to the medications, the progression of the disease, the speed at which they respond, the type of medications that they respond, the diet that they need. And it's really all individualism. And I do hope for a day where most medicine is individualized at that level, right down to the cellular level, so we understand what the person needs. And so it's a day that, that I hope for and pray for, especially in the cancer world, on behalf of my patients, but also for myself and my family and you and your family, Heather. I want that so bad.
Yeah.
Another question for you. So, what would you tell folks tell other doctors? Say you find someone with solitary fibrous tumor, you find someone with an ultra rare disease. I think as doctors, you're like, boy, I've never even heard of this. So I'll send you to an oncologist. But a lot of oncologists have fairly limited experience with this particular one just because it's so rare, and other rare cancers find themselves in that same boat. What would you advise physicians to do when they find someone with an ultra rare cancer diagnosis?
Yeah, I would say listen to the patient, because I am unique in that I have the background to understand the science. And I've done a lot of research, but I feel like a lot of people with rare diseases, they do a lot of research. They look up all this information. And so if you just start by listening to what the patient knows about the disease, then that can maybe trigger some ideas and some therapies that aren't standard, because there typically isn't a straightforward answer. I know I do that with a lot of the SFT patients online, on the Facebook forums and whatnot. A lot of them come to me and they're like, I have this new disease in this new place. What should I do? How should I treat it? So I just tell them what I think. I think that's all that patients are trying to do is just find answers and alternative approaches so they don't end up a statistic. I know that some physicians could be a little bit, not really intimidated, but they see the patients as arrogant, or potentially like coming across like they know something and they haven't gone through all that training, the 8 years or whatnot. But I think some of the most experienced and best treatments I've received are from doctors that just take the journey with me, and they're willing to try. And in addition to my liver being treated more times than anyone that has ever been treated in the world, in my brain I've had 140 SFT lesions treated by gamma knife radiation.
Your story has been wild. Wow.
He took that journey with me, the radiation oncologist. Like, we get him on that 3 millimeters, and radiation these days has developed so focused and precise that you can treat a lesion and it goes away. If you look at my brain scan, it looks like a fairly normal brain, like a brain scan, like no lesions or anything in there. He also, Dr. Warnock at UT Southwestern, he's like, we've never treated this many lesions in the brain before for a clinic. He's willing to take the journey. I'm willing to take the journey. And so far, we've been fairly successful. I know at some point I'm hopefully going to develop a cure. But until then, this approach has been a game changer.
Yeah. No, I like that. Just thinking about what you said, I think it's much more meaningful for me as a patient. It sounds like for you as a patient, when the physician is ready to go on a journey and doesn't have a predetermined thing that we are going to do no matter what, especially on rare stuff like this. There's a lot of things that you research and understand and know, and your physician may understand a lot about cancer, but they may not know much about this particular cancer. So that joint decision making and advocacy on behalf of a patient, I think that makes a huge difference when you know you're heard and listened to. And you've been blessed with physicians who have done that for you. And that's quite a gift to find people who are willing to do that, and also to take the time, because it takes more time to listen and go on these journeys rather than just to prescribe whatever's kind of the quick standard of care. It's never quick, but it's a more thoughtful journey if you have to go and explore and advocate. And then if you don't live by a place that does some of these more advanced or more, I guess, just cutting edge type things, you may have to send people to travel to those places or contact colleagues in those locations. And it sounds like you've been blessed with doctors who've been willing and able to do that.
Yeah, I'm only here because of the doctors. I say I'm paving a new way, but if the doctors wouldn't agree with it and see some merit in it, and suggest ideas and things like this, then yeah, I don't think I would be here.
Wow. That's a miracle, Heather. I just hope it keeps being successful and that we keep finding more and more answers. The more people that look into it, the more awareness that comes, the more people that research it, the more we'll learn. We can do things collectively as a group when we put our minds to it. And it takes money, it takes resources. But like you said before, I don't think that's money poorly spent. I think those things are broadly applied to many different cancers. The more we learn about it, the more useful it becomes. I'm thinking back to my undergraduate. I had a degree in microbiology, lots of molecular biology. And at that time, there was a lot known. But since that time, CRISPR technology came out, and there's just so much more like messenger RNA, being able to modify that and deliver therapeutics that involve that technology. I just feel like it's exponentially grown, even in the time since I was in my undergraduate degree. I think it's fascinating, but also has very real world applications. And it's neat that you've been able to mix the two of those things.
Yeah. And I had a core biopsies in a calf muscle SFT, and I told the physician, I was like, if one of my grad students comes up and we got the IRB stuff approval and everything, could we get some sample? And she was like, I'm going to cryoablate this tumor. Do you want to do research or do you want to get treatment? And I was like, both.
Both.
She core biopsies and then give them to my grad student who drove 5 hours back to the lab and trying to start a patient-derived cell line. And then she finished off the cryoablation to the rest of the tumor. Just doctors that are open to furthering science and but also providing patient care. And yeah, it's really great.
Yeah. So what would you tell someone who is a medical student right now? Maybe they're an MD, PhD student, and they're thinking about getting involved in this. Like, you've been able to stay in this despite serious health challenges. And I think a lot of people would have just left a career at that point or tried to live on whatever disability insurance they might have had. But you've stayed engaged and focused, which I think is amazing and miraculous. I want to know, what would you tell someone who is an aspiring researcher, clinician, especially those that are pursuing that MD, PhD path, or they're a PhD candidate looking for opportunities to do clinical research? What advice would you give them as back over time?
Yeah, it's really a personal choice, what kind of research, PhD side of things. I have a lot of friends who have done an MD, PhD. Decision after they get their degrees is sort of, well, do I want to go to practice or do I want to be a researcher? Because if I want to be a researcher, that's a lot of work. You have to apply for grants, you have to get funding, you have to commit to it. And then if you want to do clinic, that's really demanding too, patient care. And so it's really hard to do both fully and successfully. So my advice would just be to find a way to sort of merge the two, where you're seeing patients of a certain type of disease that is, you have a personal connection or some kind of drive to solve that. And then if your hospital is fortunate enough to have a clinical lab, then you can get patient samples and do research in the lab and really kind of do both efficiently and effectively. So just start thinking about that, how to make those two merge together, because it will keep you from the burnout that you talk about and just trying to juggle multiple things and not having time.
But I don't think you would still be doing what you're doing, number one, if you didn't have good clinical care. But number two, I don't think you would keep doing it if you weren't passionate and driven to solve this for yourself and for others. So in the deepest, most sincere way, I can say thank you, Heather, for what you're doing on my behalf and on behalf of so many patients who have this disease and others. I love the passion and the drive that you've exhibited. I think that's really good advice for those who are coming on board. If you find something that you love and you're passionate about, find a way to strike that balance so it doesn't overwhelm your life. You can find such a successful career. And anyway, I've loved this discussion so much, Heather, in total admiration of what you're doing, and just appreciate it so much. Do you have any final words or advice you'd want to share with our audience?
I mean, I just want to say thank you, Peter, for being the voice and letting different physicians, I've been listening to them since I've met you. And yeah, it's so amazing what doctors can do with not only their skills and their experiences, but also just as a good human being going out into the world and just helping people and developing things and seeing needs and solutions and the support that we get from helping each other. So it's really inspiring. And thank you, Peter, for facilitating all this.
Thank you so much. Heather, keep in touch. I'll be very interested to hear your story and your journey. And of course, I'm very interested in your research as well. So we'll be in touch, and I appreciate it so much. Thank you very much.
Thank you.
Thanks for tuning into the Doctors Making a Difference podcast. And thank you for what you do to help your patients and your community. Your work truly helps so many people. We produce this content to help you have the tools you need to stay in medicine and to highlight the amazing work being done by physicians around the world. Please note that while I am a physician, and many of the guests on this program are also physicians or other professionals, the discussions on this podcast do not represent my employer or any professional organizations to which I belong. This podcast is for your information and entertainment only, and should not be taken as professional advice. You should seek appropriate professional advice pertaining to your own situation. Please check out more of our content on our website, doctorsmakingadifference.com. Also, please follow, like, or subscribe on your podcast player or YouTube channel, and give us a 5-star rating. It really helps to spread the message. Finally, if you'd like to be a guest on the podcast, or if you'd like to nominate someone else to be a guest, please visit the website or email admin@doctorsmakingadifference.com. See you next time.